The cell cycle is divided into two major phases: interphase and the meiotic phase, otherwise known as the M phase. In the interphase, the cell grows and makes copies of its DNA. During the M phase, the cell separates its DNA, creating two sets, and divides the cytoplasm, thus forming two daughter cells. The cell spends most of its time in the interphase stage. In the interphase stage, excuse me. Some terms I want to get across before getting into this cell cycle is that the mother cell is the original cell that divides, that's doing the actual division, that's going through the cell cycle, while the daughter cells are the two cells that are product of mitosis and of this whole cell cycle system. Okay, well the cell cycle starts at the G1 phase, also known as the first gap phase. In the G1 phase, the cell grows larger and makes copies of its organelles. A fun fact I wanted to mention is that a cell doesn't always have to grow in order to divide. This is seen in cancer cells, as sometimes they divide upon completion of mitosis, and they just end up getting smaller. Now moving on to the S phase, which is the synthesis phase. This is the stage in which a cell synthesizes a copy of its DNA in the centrosome, which plays a vital role in the DNA separation during the meiotic phase. The final stage of the interphase is G2, and this is the stage in which the cell grows even more and reorganizes intercellular organelles in preparation for the M phase and for the start of mitosis. Entering the M phase, the process of cell division starts in mitosis. In mitosis, the DNA reorganizes itself into chromosomes and is begun to be pulled apart by the microtubule or spindle fibers. Mitosis is divided into four stages: prophase, metaphase, anaphase, and telophase. The M phase concludes with the cytokinesis, in which the cell cytoplasm is split, and with each half of the two copies of the cytoplasm each going into one of the newly formed daughter cells. I'm now going to show an example of mitosis in action. As you can see here, we have prophase. The cell seems kind of normal, and as the metaphase, you see the spindle fibers start to pull and pull, and this is more properly observed in the anaphase stage. As you can see, they're starting to pull more apart, and the telophase is now pretty much two cells. Which is missing is the cytokinesis stage, which at that point is pretty much two different cells already, and the complete division of the cytoplasm at that point. Now, the cell cycle checkpoints, they have checkpoints. You can see these white lines. These white lines right here, they're one's located here at G1 and another one at G2. They're just there to make sure that everything's ready to advance to the next stage, and that the cell cycle can continue on in a healthily or more in a healthy manner. However, if the checkpoints of C that the cell isn't ready to go on, it won't go on until it is ready to go on. Now, I want to talk about now is meiosis, and I have a diagram here as well, just as I had for mitosis. Move this away real quick. I'm gonna move this out of the side. Here's meiosis, and essentially what happens here. This requires the separation of the sister chromatids and the homologous chromosomes. The separation of the homologous chromosomes take place in meiosis 1. As you can see here in meiosis 1, this is pretty much it's kind of like mitosis, only they're not identical. They're like pretty much splitting up the homologous chromosomes they have like in half. And here at this point, they're split in half. And after that, it proceeds on to meiosis 2. As you can see here, it goes again, and it splits the chromatids. It splits the sister chromatids as observed here. You see 2 here, and over here at this point you just see 1 in each. So So this is an example of meiosis. Now, crossing over occurs when the homologous chromosomes exchange genetic material at random rates. Crossing over can happen at higher rates and at different times if meiosis happens more frequently in the organism as it does in humans. Well, that's the cell cycle, meiosis, mitosis, and that's pretty much it. So thank you.